Okay, so this is Medical Genetics, Lecture 2. We're going to talk about genetic variation. So Mendel was able to demonstrate, of course, that traits can be passed on, but also that recessive traits can be masked. So this is just an example of masking of some traits. And so here we have little h, and big H, and you can see that every single offspring is going to be heterozygote. So every single offspring will have a big H and a little h. On this side, you have big heterozygote parents, and in this case, you're going to have some homozygotes that are going to be produced. So traits can be passed through generations, especially recessive, autosomal recessive traits, and they don't show up for generations because of masking of those traits. Now we want to talk a little bit about the origin of genetic variation or mutations, and so we can think of this in terms of two different areas. One is, of course, the germline, what gets passed on from generation to generation, and the second is what happens spontaneously or sporadically, which is somatic mutations. So in the germline, of course, this will be passed from generations. And then somatic is going to be at the level of the individual and has a potential also for cancer, of course. Now we also have mutations versus polymorphisms. What do you want to think about polymorphisms? Polymorphisms are allowed mutations, you can think of it that way, whereas mutations in the original sense means that you've changed the gene in some way. Typically, either it has either enhanced function or a loss of function, whereas a polymorphism is a change in the gene that gets passed down and is inheritable. And oftentimes with polymorphisms, there might be some sort of evolutionary pressure for that polymorphism. So mutations are very rare. They're going to be in less than 1% of the populations. And classically, polymorphisms are considered to be in more than 1% of the population for them to be considered a polymorphism and stable. That definition of polymorphism modern is a little bit wiggly, and so polymorphisms now can have a range of less than 1% to greater than 1%. And this is a more modern take of polymorphism. Now, spelled modern wrong. So, but mutations are still rare. Okay, so these are going to be rare. And likelihood the mutation is not going to be favorable and oftentimes doesn't lead to a viable offspring. So we have different types of mutations, and I'm going to go through several of these mutations and just talk about what they are. So the first mutation is a base pair mutation. Okay, base pair mutations is simply that you just have a single base pair that gets mutated. We've talked about this with respect to sickle cell anemia. Now there are different types of base pair mutations. You can have a silent mutation, and this occurs when the actual change of the base pair doesn't alter the amino acid. So remember, we have redundancy within the code, and so some amino acids have multiple different codons, and so a silent mutation will generate the same code. Will generate the same amino acid using a different code. A missense is what we saw with sickle cell anemia, in which you have an altered codon with different amino acids. However, the codons are still going to be grouped together with amino acids that have similar properties. So oftentimes a missense mutation will lead to a mutation but with an amino acid with a similar property, a negative charge for a negative charge, positive for positive, etc. And then you have a nonsense mutation. A nonsense mutation is important because it introduces a stop codon. And the stop codon is going to more than likely produce a non-functional protein. And so this is referred to as a truncated protein. And truncated means that it is shortened. Okay, so the natural protein is shown here, the shortened truncated protein is shown here, and it probably is not going to fold properly, and it most likely will not be functional. Now, another type of mutation is going to be a deletion, and deletions typically occur during the process of replication, where a mistake is made that is not fixed, and you get removal of nucleotides from the actual genome. So you lose this area, so this is lost DNA. And this lost region could be small. It can affect one gene or it can affect many genes. Some examples of this is Turner syndrome. And in Turner syndrome is when you have a full chromosome that is missing or partially missing, and that chromosome is the X chromosome. So this is only present in females. More likely, if it did occur in a male XY, it would be a non-viable offspring. But because the X chromosome, you have 2 in females, this is a viable offspring or viable fetus, but it will have some effect on the fetus, and those are referred to as Turner syndrome. Cystic fibrosis has also been associated with deletions, and it has variability in what genes are deleted. So cystic fibrosis is a very heterogeneous disease. Williams syndrome is a deletion of chromosome 7 region, and it usually includes 26 to 28 genes. People affected with Williams syndrome are usually confused with Down syndrome. They have some similar features. Down syndrome is more common than Williams syndrome. But again, Williams syndrome is a deletion or loss of genes from that chromosome. Another type is insertion. Insertion is going to be where you are going to put genes in a different location. And so your insertions can change the proteins that are going to be produced. So you can insert in between genes, in the middle of a gene, or outside of a gene, etc. So it just depends where you insert. And it's going to insert wrong amino acids, which can lead to early termination of genes that has been interrupted. It can change your genes to nonsense mutations in which you have a non-functional protein. It can also move a potential oncogene to a strong promoter, and this can lead to cancer. So moving a gene that is not expressed to very high levels next to or near a promoter that is expressed at very high levels has potential for cancer formation. Another type of mutation is a frame shift mutation. And an frame shift mutation is that when you get an insertion or deletion of nucleotides that's not in multiples of 3. And this is the most detrimental mutation. So here you can see is the normal gene, and you get the normal amino acid sequence shown here. You insert one amino acid, and that shifts the frame. So now these last 2 gene or amino acids are different, and this will lead to a different sequence downstream as well. And then you can also have an insertion of 3 trinucleotides shown here, and this will only change one of the amino acids, and everything else will be the same. So that is a non-frame shift mutation, and so that will not be as deleterious as a true frame shift mutation. The frame shift mutations can be in numbers of 1 or 2, anything that disrupts that 3. In addition, you can change the stop codon when you do this, and this will lead to either a longer or shorter polypeptide, which of course will affect its function as well. Now, other types of mutations are going to be mobile elements. Mobile elements are going to be pieces of nucleic acid that can in themselves be moved around. And we have, for example, transposons, and these can insert themselves into DNA, and they are mobile elements. And so they get cut out, and when they get cut out, they take with them a section of DNA, and then they're going to insert that section into a target DNA. Now, this is commonly used in bacterium because bacteria will move around antibiotic-resistant genes in this way, where they're going to take, using transposons, they'll take an antibiotic-resistant Staph aureus, and they'll transfer that to a plasmid that can then be transferred to another Staph aureus to make them antibiotic resistant, for example. Another type of mutation is a duplication. Duplication is just what it is. You get double the material. Typically, this will result in more product being made. It can, depending on where it's duplicated, interrupt genes as well. But more commonly, it's just you're doubling the genetic material, and so you can potentially double the amount of protein that is within this region of the genome. Now, another type of mutation is referred to as a splice site mutation. So we know what splice sites are. They are to remove introns so that your exons can come together and form the messenger RNA that will ultimately be the protein. So, for example, we have a gene here in which you have exons. So you have exon 11, exon 12, and exon 13. And normally you get spliced out your introns, and your introns are intron 11 and intron 12. And then you get your messenger RNA, which has all your exons with your introns removed, and it leads to protein A, for example. Now, in a splice site mutation, it can be such that you change the actual splice site. So you can see here that we had a change from a guanine to adenosine, and this altered the splice site so that now you're going to remove intron 1, exon 12, intron 12, and you're going to get a altered messenger RNA, which is missing exon 12. So this is going to be deleterious, obviously, to the actual protein. And this is something that is seen with PKU, which is a deficiency of what's called phenylketonuria, and this is going to be prominent in European descent. And basically, it's just that you are changing the sites from that G to that A in the intron 12. And so this is now not going to be recognized by the splicing enzyme, so it's going to be bypassed. And this bypassing allows exon 12 to be lost. And so you get a truncated protein, which is unstable and has no activity. There are also promoter mutations. And promoters, remember, are upstream of the gene, and they not only contain the binding site for transcription factors, but can also contain binding sites for regulatory factors. So the mutations in the promoter region, if it changes the level of transcription, then that indicates that that mutation is altering some sort of binding site. And again, it could be a binding site for transcription factors or regulators. Another type of mutation is an expanded repeat mutation. And so this can lead to what's referred to as a pre-mutation or carrier that gets passed on from generation to generation. This is seen with Huntington's disease. And each generation, the repeats get larger and larger until they actually present with the disease. And so a person who is the original carrier might present with a Huntington's disease at the age of 60, and their children, because they inherited the expanded repeats from their parent, they can present with Huntington's disease at a much earlier age because they start with the pre-mutation. Oftentimes these pre-mutations are going to be outside of the genes. So in the case of Huntington's disease, again, it's not occurring actually within the exon regions, but it's occurring upstream of that. And just by adding more and more mutations upstream, you make it so that the gene can no longer be properly expressed. Some can be encoding regions, however. There are several that are going to be linked to neurologic diseases. And the most common repeats for neurologic diseases are CGG and GCC repeats. And so this is just showing in a normal protein, you have a CAG repeat, which is going to be about 5 to 35 repeats. That's going to be normal functional. And then as you get expansion of these repeats through generations, this now becomes not a coding protein, or it affects the coding of the message for the protein. So there are some examples of trinucleotide repeats, and disorders associated with central or peripheral nervous system often have these repeats. So you have a type 1 trinucleotide repeat, which is CAG, and so you're going to have glutamine expansion over and over and over again. And in these genes for the central and peripheral nervous system, about 20 repeats is normal, and then if you have more than 20, you go into what's called a polyglutamine disorder. A type 2 trinucleotide repeat disorder will be made up of variable trinucleotides from CTG, GAA, GCC, CGG, and these are all within non-coding regions. And so if you have, for example, Fragile X syndrome type A, it's going to be an expansion of GCC in the 5' end of the non-coding region. If you have Fragile X syndrome type E, it's going to be CGG at the 3' end. Myotonic dystrophy is going to be a CTG at 3' end, and Friedreich ataxia is going to be GAA within an intron. So again, you don't have any of these occurring actually in exons. They're either at the 5' end, the 3' end, or within an intron that are causing the disease. Okay, so overall consequences of disease or mutations are going to be a gain of function, a loss of function, or you have a dominant negative effect. So a gain of function is going to be dominant, typically, and you can lead to a novel protein or overexpression of a normal protein. Or inappropriate expression. It's not supposed to be expressed, and it is being expressed. A loss of function mutation is usually recessive. And you're going to lead to situations in which you're not producing enough of the protein. Depending on what the protein is, it will lead to either a very strong phenotype or a mild phenotype. So you can have 50% loss of protein, and this will lead to what's called haploinsufficiency. And so haplo just means half, and insufficient means you're making half of the protein, so it's an insufficient amount. In a normal individual, you make 100% of whatever the protein was, and so by losing that ability, it's referred to as haploinsufficiency. An example is the LDL, low density lipoprotein. So if you have a loss of function of low density lipoprotein, this is going to increase cholesterol in the blood. And it's because your low density lipoprotein is going to help your high density lipoprotein remove cholesterol and bring it to the liver for the liver to dispose of it. There's also dominant negative mutations, and this is when you have an abnormal protein and it has a dominant effect on the normal protein. Okay, and one example of this is our prion protein. Even though you make normal prion proteins, if you have the mutated version, it's going to have a dominant effect over the unmutated. Now, an example of a genetic disease is Achondroplasia. This is where you have your gain of function mutation of a tyrosine kinase receptor. So the gene is this FGFR3, and it is a transmembrane tyrosine kinase receptor. We do know of tyrosine kinase receptors. Insulin has one, for example, and these typically are going to be associated with growth. And FGFR3 is also associated with growth. It's going to bind fibroblast growth factor, and its signaling is going to inhibit proliferation of chondrocytes within the growth plate. And so this is FGFR3 is going to help coordinate the growth and differentiation of chondrocytes. Now, you might think that it's a binding the growth factor, so it must promote growth, but that's actually the opposite of its function. It's a regulator of growth. And so it's going to inhibit proliferation. So a gain of function of this particular gene would result in a decrease of growth, right? And so if you have a gain of function mutation, this missense mutation, it's going to affect the growth of the chondrocytes, and so you're going to have a shorter stature, for example. And so achondroplasia is associated with human dwarfism. This is an autosomal dominant disorder. The mutation is at guanine of position 1138, which is seen in 100% of the cases. 80% will have the de novo mutation in the father's germline, and the frequency increases with advanced paternal age. The onset is going to be prenatal, and you're going to have that short stature associated with it, as well as megaloencephaly and spinal cord compression. Right, so there are also a series of hemoglobin disorders. And so just as a review, we have hemoglobin A, and this is going to be composed of 2 beta chains and 2 alpha chains. And this is going to be normal hemoglobin. And we also have fetal hemoglobin. And fetal hemoglobin is 2 gamma chains and 2 alpha chains. And then we also have your hemoglobin A2. And hemoglobin A2 is going to be 2 delta and 2 alpha chains. And so there is a common theme here. You have your 2 alpha, 2 alpha, 2 alpha. So that should tell you straight up that alpha is very important. Now, one area where you don't have alpha is embryonic hemoglobin. Embryonic hemoglobin is where you have 2 epsilons and 2 zeta. I'm not going to try to draw the zeta. So for fetal hemoglobin A and embryonic, these 3 together are going to make up about 3% of normal hemoglobin. And then, of course, hemoglobin A is going to make up 97 to 98% of all your hemoglobin. Now, you have different locus. We have what's called a beta cluster, globulin cluster, and an alpha globulin cluster. And so the beta cluster is on chromosome 11. And then the alpha cluster is on chromosome 16. Okay, and so with respect to your beta cluster, you have your epsilon, which is part of your embryonic. You have your gammas. You have your beta. You'll have a delta. And on chromosome 16, you have your alphas and you have your zetas. And so these are just, again, these are clusters of globulin genes. And so if you get mutations in the globulin region, it could affect one or more than one of these genes because they're clustered together. So other thing to keep in mind is that you have 2 chromosome 11. So you have a maternal and you have a paternal. And also chromosome 16, you have a maternal and you have a paternal. Now what that means is there is one beta region, so you have a maternal beta and you have a paternal beta, which means you have 2 beta genes. Now for the alphas on the maternal, you have 2 alphas and 2 alpha on your paternal, so you have the potential for 4 alpha genes that you inherit. Okay, so hemoglobin disorders have different categories. We have our alpha thalassemias and our beta thalassemias, and the thalassemias are a structural deficiency or defect. And so you can have alpha thalassemia or you can have beta thalassemia. And so what this thalassemia is the actual molecule is going to be altered. So it's going to have a structure that's normal, but is not going to have normal expression. You have, in the case of thalassemias, you can have hereditary persistence of fetal hemoglobin, and that can compensate for the lack of your normal hemoglobin. So thalassemia is a reduction in either alpha or your beta chains. So here you show your beta chains and your alpha chains. And then, of course, this will make up hemoglobin, in which you're going to transport oxygen, for example. So you have homotetramers are going to form when one chain decreases. So again, hemoglobin A and B, you want them in balance. And if one chain is mutated, then you're going to get more of the other chain. And homo, of course, means same. So you'll have 4 alpha chains or 4 beta chains, for example. If you have a beta or a gamma homotetramer, you have decreased oxygen binding, and this can lead to hypoxemia. Alpha homotetramers are going to precipitate, so they tend to come out of solution, which your blood is a solution, and so they're going to damage your red blood cell membranes, and this is going to lead to edema due to your loss of red blood cells due to the integrity of their membranes. So some clinical consequences of our thalassemias are as follows. We have alpha thalassemia syndrome, and these are going to be associated with that chromosome 16. If you have 2 genes that are mutated, you have what's called thalassemia minor. The hemoglobin electrophoresis is going to be normal. If you have 3 genes that are mutated, this is going to be referred to as hemoglobin H disease. You're going to have hemoglobin H, which again is that beta homotetramer. So your alpha chains are replaced with beta chains, and this leads to splenomegaly. If all 4 are going to be mutant, you have hydrops fatalis. This will lead to hemoglobin Bart's disease, in which you have the gamma 4 tetramer, and that will be fatal. Now, beta thalassemia, again, this time you only have 1 beta gene, so you only have 2 genes, one from your paternal chromosome 11, one from maternal chromosome 11. If you have 1 gene that gets mutated, that's called beta thalassemia minor. If you have both beta chains mutated, this is beta thalassemia major, or Cooley's anemia. Complete absence of beta chain is beta 0 thalassemia, and this is usually associated with a nonsense or frameshift mutations. If you have a reduction of beta, you have 10 or 30% reduction of beta, is a beta plus thalassemia, and this is usually associated with a locus control region mutation or a splice site mutation in either the introns or the exons. This is just an example of beta thalassemia major with splenomegaly. And splenomegaly is present again because your spleen is where you're going to filter your blood and remove blood cells. And so because you have so many deficient red blood cells, they accumulate in the spleen and your macrophages get overwhelmed with trying to remove them. So beta thalassemia syndrome is a loss of function mutations. And with respect to these mutations, people can be what's called a compound heterozygote. The compound heterozygote is the presence of 2 mutations that are different of the same allele. So our allele 1, for example, has a mutation at a different location than our allele 2, and this is a compound heterozygote. Whereas in our other individual, you have mutations at the same loci in the same gene, and this is called a homozygote. Now we talked about sickle cell anemia. So sickle cell anemia is that single base pair substitution in the beta globulin chain in which you get valine for glutamic acid. So you're switching a change of amino acids. And not only that, you're going from a more hydrophobic to a hydrophilic. And this is, of course, going to be a mutation that changes the amino acids, and you get a cyclic or a, I did that wrong, I'm sorry, thought I was doing this wrong, hydrophilic to phobic. Sorry. So your valine is hydrophobic and it wants to hide from water, and so it sort of pushes the red blood cell membrane inwards so it can hide, and that causes this sickle cell shape. Now the consequences of sickle cell anemia, of course, is that you're going to have cells that are going to get stuck, especially as they go through capillaries. So it leads to vascular obstruction, and this can lead to hypoxemia in tissues in which they're not able to get oxygenated blood supply, and this can lead to an infarction or death of those tissues that are affected. You also have destruction of reticulocytes, and so this will reduce the amount of blood cells, reduce the amount of hemoglobin, leading to anemia. You can also have the spleen that's going to destroy many sickle cells and get an enlarged spleen. This can lead to death in the spleen if it's overwhelmed, so that's the infarction. It also can lead to suppression. The spleen is a very important organ with respect to your immune response, and so individuals have an increase of bacteria infections. So for protein electrophoresis and identifying individuals with sickle cell, you can see how they're different by putting it on a gel in which you have a current go through, and you have one end of the current is negative, one end is positive. So you have your hemoglobin A, which is our normal hemoglobin, and it kind of goes in between positive and negative. And then you have your hemoglobin S, which has that hydrophobic, and so is more attracted to the negative end of the gel. And then you have your hemoglobin heterozygote, which has a band for the hemoglobin S as well as the band for the hemoglobin A. Okay, so treatments for these types of diseases are going to be blood transfusions. You're going to also have chelating agents. Antibiotics are going to be used because, of course, you're going to have individuals more susceptible to bacteria. And so you kind of have them on pre-exposure prophylactic bacteria. Your chelating agents are going to help bind to ions. And so that's going to help separate because you have a lot of your destruction of your red blood cells, and so you're going to have a lot of hemoglobin. Chelating agents will help to make sure that the hemoglobin is going to be broken down by your liver, for example, and not be picked up and used for other reactions. Vaccines are important, so we have a pneumococcal vaccine that's going to be important again because of susceptibility to bacteria. A lot of this is associated with pain. You can imagine that if you have red blood cells getting caught in your capillaries, your nerve endings are there as well. And so people are going to have to use analgesics for a sickle cell crisis. Bone marrow transplants, unfortunately, that's associated with high mortality rates in sickle cell patients. There is some drugs that are being developed to target fetal hemoglobin, try to reactivate, for example, that gamma gene. And also there is some gene therapy for beta thalassemia. All right, so now some causes for mutations. Again, you can inherit a mutation, but most of them are going to be spontaneous during DNA replication. We, of course, have fidelity within our DNA, so we have some repair mechanisms that we'll go over just in a moment. And so you have also, of course, environment and mutagens that you can be exposed to, and these are going to have an influence on mutation as well. For environment, you have ionizing and non-ionizing, which we'll go over, and of course, repair. So for ionizing radiation, these have the potential to break your DNA. They can alter your bases. They can actually get in pretty deep and reach germline cells, your eggs and sperm. An example of this are X-rays. Non-ionizing are going to be those that are going to have an effect on electrons and making them, or putting them in the outer orbit so they're more reactive, and that makes them, the atoms that they're part of, very unstable. These are going to cause covalent bonds between pyrimidine bases, and they're going to cause base pair substitutions, dimer formations during replication. Non-ionizing cannot reach the germline, and UV radiation, sunlight, is an example of that. So here's an example of UV radiation. So you have ultraviolet light, and you have a thiamine dimer. So here's our thiamine dimer. And so your ultraviolet light is going to cause a mutation, cause the dimer and the base pairs near the dimer to be removed. And so your cells have to repair this. They have to come back in and repair and replace and put those sequences back. So just as a side note, some people are always thinking that radiation, what happened with our atomic bombs and stuff, are going to affect multiple generations. And so there was a study done on 76,000 offsprings of Hiroshima and Nagasaki bombing. And so this is looking at the germline and the passing down of mutations to offspring. And they also did a study on children exposed to Chernobyl nuclear power plants. And so here they're looking at not only germline, but somatic mutations. Was there an increase in somatic mutations? And of course, this is in comparison to normal populations. And so the finding was there an increase in somatic mutations? And surprisingly, no. And was there an increase in germline mutations? And there was not. So what does this mean? Well, this means that our body does have a lot of repair mechanisms for those potential somatics. So it's pretty stable, our replication. And then with respect to germlines, our germ cells are very protected from mutations. And so that is why genetic mutations are very, very rare. Now, another thing to consider are carcinogens. So carcinogens are, carcinogenic is a mutation that increases the risk of cancer. There are very few chemicals that are true mutagens. However, those carcinogens that can increase the risk of cancer, 90% of them are mutagens. So they are going to have the ability to mutate the genome. So there's a strong relationship between carcinogen and mutagen, and this is going to be measured by a test called the Ames test. And the Ames test is used as a screen for potential carcinogens. And so just as an example of what the Ames test is, the Ames test is where you're taking a bacteria strain. In this case, you're just using Salmonella. And Salmonella is going to require histidine, so that's an essential amino acid, so you have to add essential amino acid. And so what you basically do is you take a liver extract, and they use rats, and you are going to add Salmonella to that liver extract. And then you test a possible mutagen, so you add a mutagen as well to that liver extract. And then you put on there a media with minimal histidine, and then you get a high number of revertins from histidine negative to histidine positive in the bacteria that had a mutagen present. So this would suggest that the mutagen is causing the mutations and the selective pressure of Salmonella to change from histidine negative to histidine positive. Whereas in the control plate, you have some natural revertants, but not a lot. So some other chemical mutations are going to be base pair analogs. And so these are going to resemble bases, and they can be substituted in DNA during replication. Base pair analogs are going to be used as antiviral agents against different viruses. Remdesivir, for example, is a base pair analog that has been used for hepatitis C and Ebola, and now it's been being tested for SARS-CoV-2. Acridine dyes can incorporate in between bases. They don't really alter bases. However, because they incorporate when the DNA does replicate, it can cause a shift, a base pair shift, or a loss of bases just by the presence of those dyes. Nitrous acid can remove an amino group from cysteine and convert it to uracil. And then uracil is going to pair with adenine instead of guanine, and so you get a base pair substitution. You also have mustard gas, which is a very powerful mutagen, vinyl chloride, formaldehyde, which is used commonly to preserve tissues for study. You have saccharin, which is a weak mutagen, but it has been shown to be a mutagen, and that can be found in some of the artificial sugars. Aflatoxin B is a fungal product, and that's going to contaminate food, and it's a natural mutagen. Aflatoxin B is going to be processed by our P450 enzymes, and our P450 enzymes will actually make aflatoxin B more toxic by a chemical reaction. Now we also have, of course, DNA repair. So we want to maintain the fidelity or accuracy of DNA. And so this fidelity is going to be maintained by numerous enzymes. And again, those checkpoints during the cell cycle are where they're going to say, do we need to move up through cell cycle, or should we undergo apoptosis? We talked about this in biochemistry. In order for DNA to be repaired, your repair enzymes need to be able to identify the parent strand, because you don't want them to change the parent strand based on the mutated daughter strand. So in bacteria, they methylate the parent strand. The mechanism in humans of how our repair mechanisms identify the parent strand is not known. So E. coli has many different repair mechanisms. There is a mismatch repair, nucleotide excision repair. This is going to be to remove that pyrimidine dimers, and this is can lead to a disease called ectoderma pigmentosa. Direct DNA break repairs, and this can lead to some cancers. So ectoderma pigmentosa is where you have faulty DNA repair, and so UV radiation will naturally cause pyrimidine dimers in the DNA. We talked about that. So you need that excision repair to remove the dimers and then repair and put back the base pairs. This is an autosomal recessive disease, and in this case, you have your nucleotide excision repair system does not work. The DNA dimers result in base pair substitutions, and the disease severity differs. Symptoms can appear within the first couple years of life, and often they're going to present on the skin because that's the most commonly exposed tissue to the sun. So there are various diseases associated with repair defects. So again, ectoderma pigmentosa is going to have that nucleotide excision repair defects, and this can lead to skin tumors, photosensitivity, neurologic abnormalities. Cockayne syndrome is going to have a defect in the repair of UV-induced damage, and this can result in reduced stature. We have an anemia, which is going to have different genes involved, but it's all involved in DNA repair. Exact function of those genes is not known, but you can have anemia, leukemia, etc. Bloom syndrome is going to have mutations in the RECQ helicase. So RECQ helicase is going to be a common source of mutations that leads to these diseases. So for Bloom disease, it can lead to an immunodeficiency, chromosome instability, and growth deficiency. RECQ is also going to be mutated for Werner syndrome, and this is going to lead to cardiac, skin elasticity, short stature, etc. You have an ataxia telangiectasia, which is going to have a problem typically in halting the cell cycle after DNA damage. So those checkpoints, those p53s, are not going to be able to prevent progression to cell cycle if the DNA is damaged. Hereditary non-polypose is a mutation of 6 DNA mismatch repair genes, and this is going to lead to a propensity for cancer. So there also are a place where size is going to play a role in mutation rates. Duchenne muscular dystrophy gene, for example, is 2 million base pairs, and of course, it's going to have introns intervening those base pairs. The somatostatin gene is only 1,480 base pairs. So you have a more likelihood of having a mutation in your muscular dystrophy gene than you do in your somatostatin gene, for example. Mutation hotspots are CpG motifs. 80% of CG dinucleotides are methylated, and in this case, the methyl group is going to be attached to cytosine. Mutation rates also increase with age, and so you have a relative incidence of mutations, and these are different diseases associated with somatic mutations, and they increase as you age in your population. We also, of course, have our ABO antigens, and these were discovered in 1900s. They were identified as being a cause of what was referred to as a hemolytic reaction following transfusion. ABO refers to the presence or absence of the antigens. So you have different genotypes associated with your red blood cells. You have an AA, which you have a blood type of A. You have an AO, in which you have a blood type of A, B, BO, AB, O, which is the absence of A or B. And then you have your rhesus antigens. Rhesus antigens were identified in rhesus macaques, a type of primate, so that's how they have the RH. And then it was found that these RH factors are also on human red blood cells. So you can either be Rh positive or Rh negative. Now what these blood types mean is if you're blood type A, that means you will mount an immune response against the B antigen. If you're blood type B, you will mount an immune response against the A antigen. And if you're O, if you're AB, you have no immune response to either A or B. If you're O, you will have an immune response to A or B. So what this means is that the O, of course, is going to be the universal donor, and the AB is going to be the universal acceptor. So there are some methods to detect variations at the DNA level. One such method is Southern blotting. Southern blotting will measure DNA. There are two other blots just to mention. There's Northern blot, which is RNA, and then there's Western blot, which are proteins. And so all these blots are doing is basically you have a gel and you're going to run in this case it's Southern, so you're going to run your DNA on the gel, and they're going to migrate according to size. So the larger fragments will be more to the top, higher, and the smaller fragments will move through the gel much faster. And so you have restriction enzymes that will cleave different sites of the DNA. So what this is showing is that you have what's called an ECO-R1. This was found in E. coli. That's where the ECO comes from. Restriction enzyme 1. And the restriction enzyme 1 can identify this sequence, GAATTC, So anywhere in the genome that you have this GAATTC, your EcoR1 is going to split that or cut it. And so you get fragments of the DNA, and then you can run the fragments on this blot, and the fragments will be shown or identified using probes, and they will glow, and then you can identify different fragments on the plot. Okay, so southern blotting is going to be important because it's going to be able to show different proteins. So here we have different samples A, B, and C, and they have different proteins that are going to be visualized, and you can see these using X-ray films. Okay, so within any population, you're going to have what's referred to as population variation, the Hardy-Weinberg principle. The Hardy-Weinberg principle is going to specify a relationship between gene frequencies and genotype frequencies, which is going to be useful in estimation and prevalence of disease. And it's also useful in estimating the incidence of carriers of recessive alleles. So again, we started with this lecture on talking about how recessive alleles can hide within a population. And so these estimations are important for determining the incidence of carriers of these alleles. So here, of course, we have our A, or gene A, which has big A, little a. And so you have a little a, which is a homozygous recessive, and of course a big A, which is a homozygous dominant gene. And then you have your heterozygotes. And so the Hardy-Weinberg variance population would estimate that in order for you to have a little a, and a big A, obviously, you have to have heterozygote parents. And so that's sort of the principle, that's how it would be used. There are also other causes of genetic variation due to natural selection. So natural selection is going to have some sort of pressure for a mutation to be more stable in a population. And this just shows that you have a frequency of the sickle cell gene. And you can see that in the sickle cell gene, you have 10 to 20% of the populations in these purple areas, 5 to 10% in these green areas, and then 1 to 5% in these shaded areas. And it almost exactly overlaps the distribution of malaria. So there is some non-overlap in this area here, but for the most part, the pressure for sickle cell trait aligns with the endogenous levels of your malaria parasite. And when I say sickle cell trait, we have our sickle cell gene. So you have your hemoglobin A, and you have your hemoglobin S. So if you are a homozygote A, you are normal. If you are a homozygote S, you are sickle cell, and this is the disease. But if you are a heterozygote, you are sickle cell trait. And it's really this heterozygote that is protected from malaria and is also doesn't have the disease associated with sickle cell. So other causes of genetic variation, we have genetic drift, we have bottlenecking, and we also have the founder effect. So genetic drift is your basically your gene frequency will deviate from one generation to the next in small populations. This is associated with uncommon genetic diseases in a large population, but it may be frequent in a smaller population. So basically the gene is going to get more concentrated if the population is smaller. So some examples of this is the Ellis-Van Creveld syndrome, which is polydactyl, reduced stature, congenital heart defects, and this has a higher frequency in the Amish population. Bottleneck is also where you have a group of individuals that are going to have inter-family relationships, and so the genes get bottlenecked to such a way that especially autosomal recessive traits, have an increased frequency. So Tay-Sachs, Gaucher's disease, torsion dystonia are all going to be bottlenecked in Jewish populations. There's also the founder effect. This is a special case of genetic drift. In this case, Finland is often the example for this. About 100 generations ago, so we're going back several hundred years, the Finland population was populated by a small number of founders. So they broke away from mainland Europe and went to Finland and founded it. And so it was just a small number of founders. So you sort of got a founder effect. It's kind of a, it's not a bottleneck in the same sense as the Jewish populations, because Jewish populations are deciding to marry interfamily marriage, whereas the founders, they are isolated, and so they have no options for marrying outside their founding population. However, what this does is that it, similar to bottlenecking, you have an increase of rare genetic diseases, 30 in fact, and then the diseases that are common in Western Europe, phenylketonuria and cystic fibrosis, are not common in Finland because those diseases came about after the Finland was founded. So the founders of Finland did not carry those mutations with them. Okay, so that's